Good afternoon, everyone. Welcome to the Data Science Seminar from the University of Washington EScience Institute. Today is a sunny day, so I'm happy that you're with us. Joining us today is Dr. François Ribalet, who is a principal research scientist at the University of Washington School of Oceanography, where he leads a team developing flow cytometers and analytical tools and mathematical models to study marine ecosystems. He first studied mathematics at the French Grand École and then pursued a PhD in population biology and oceanography with the Open University in London. François joined the University of Washington in 2008, leveraging laboratory experiments, field observations, and satellite data to build mathematical models that forecast microbial interactions across a range of ocean conditions. The title of his talk today is Ocean Warming Threatens the Survival of Key Microbes. As usual, please use the Q&A feature of the Zoom webinar version to ask any questions you may have. As part of the University of Washington, we acknowledge the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. And with that, François, please take it away. Thank you.
Thank you. Hi, everyone. First, I want to thank the organizer for giving me the chance to talk a little bit about my research. And today, I want to talk about a tiny ocean creature that you probably never heard of, and its name is Prochlorococcus. And it's really an amazing little microbe. For one, its ancestor changed the Earth. It made it possible for us, animals and humans, to evolve. And there are 3 billion of these tiny organisms in the ocean. And guys, I know you like numbers, so that's 3 octillions cells in the ocean. And we didn't know they existed 40 years ago. So I divided my talk into 3 parts. The first one, I'm going to introduce you to Prochlorococcus and tell you why everyone should care about it. The second part, I'm going to touch on some other approach I use to answer some of the questions regarding how these organisms, for instance, adapt to change in their environment. And the third part, I will show some preliminary results from a research I do on try to predict how Prochlorococcus will respond to future ocean conditions. So, as terrestrial animals, we are very accustomed to plants on land. We know about trees, plants, pastures, grass, crops, but there are 100 billions of animals in the ocean, and they are all fed by this invisible pasture that are made of these macroscopic organisms that do photosynthesis, and they are called phytoplankton. And with this phytoplankton lives also a lot of animals. They live among them, they eat them, and some other animals live in the bottom of the ocean, and they wait for this phytoplankton to die and settle down. And so here I'm showing a map of phytoplankton biomass that were made from satellite observation, where green colors show high biomass phytoplankton and blue show very low abundance of biomass. If you take all of these phytoplankton together, collectively, they represent about 1% of all the plants' biomass on land, but they photosynthesize as much as all plants on land, and including, for instance, the Amazon rainforest. They fix about 50 tons of carbon per year in the form of carbon dioxide that fed then the entire ocean ecosystem. So how does these tiny little creatures can fix as much biomass as all plants on land. Well, they don't have trunks, stems, they don't have flowers and fruits, all of this fancy equipment. They are very minimalistic. They do only two things. They grow doing photosynthesis, and when they are big enough, they divide. So this is what they do. They do two things. They grow, they divide. They grow, they divide. So they're very lean, very efficient photosynthetic machine. And so there are many different type of phytoplankton, different shapes and different size. All of them are basically smaller than the width of a human hair. So here I'm showing like an example of the most beautiful phytoplankton you can find. These are the textbook phytoplankton. We call them the charismatic ones because they're very, very pretty. And here is Prochlorococcus. Yes, not very impressive. It's very, very small, but they're there. But they are so small that you can actually not really see them with a normal standard microscope. So to kind of like give you a sense of the range, like for instance, phytoplankton size range spans 9 orders of magnitude in cell volume. And I took this illustration from a colleague of mine, Zoe Finkel, who I really like this illustration. It shows on the left, Prochlorococcus size is about 0.5 micron in diameter. And if you would compare it to the size of a fish, the difference between Prochlorococcus and this big charismatic phytoplankton I showed you before, like they are called, like for instance here, Rhizosolenia, Cerathium, Cocinodiscus, it will be the size of the Eiffel Tower. So that's the change. When we say macroscopic, yes, but you have a huge range in size among these microscopic organisms. In fact, Prochlorococcus are so small that they have been discovered by accident using a technology called flow cytometry that have been developed for medical research to study cancer cells or blood cells. And very briefly, a flow cytometer works by passing a sample through a laser beam, which excites the particles and causes them to emit light. And the emitted light is then measured by detectors, and this and which provide information about the physical and chemical property of the cell. So when people brought this technology, these instruments on research vessels to study ocean microbes, they noticed a very faint signal that first they called it noise, electrical noise, like this very, very tiny signal, but turned out to be these Prochlorococcus turned out to be the most abundant photosynthetic organisms on the planet. So now we've been, we've brought this instrument across for 40 years across the ocean. So we are now a much better understanding of their distribution. And we found them mostly in regions that are called open ocean gyre, where we see basically the red colors there. Red colors mean high abundance, and blue or cooler colors mean lower abundance. And these open ocean gyres are often referred as desert of the ocean, but they are not desert of the ocean. In this very beautiful blue deep water, you actually have 100 million cells in a liter. There are these micro. So if you would combine the biomass of all of these cells, it would be the equivalent of the weight of all human population. And they will photosynthesize as much as all crops on land. So they are tiny but mighty. And they are incredibly important for our planet. So, of course, the million-dollar question is, what are they going to do in the future? And I'm sure a lot of people have been looking into it. And it's a lot of the conclusion are based on there is a very strong correlation between the abundance of these organisms and temperature. And so with some statistics, this led to a lot of models to predict an increase of their abundance at the end of the century. And here I'm showing one of these predictions from these authors there, where they say on average, they estimate that the cell abundance will increase by 30% by the end of the century. That's, if you like me, you love Prochlorococcus, this is great news. But I wanted, 10 years ago, I wanted to go into a more mechanistic approach to make this type of prediction. I wanted to move beyond just correlation based on abundance, because this correlation between abundance and temperature, and use a more mechanistic understanding for this population. So how I'm doing this. So we know that change in biomass for these microbes is the result of growth, mortality, and migration. And for phytoplankton that live in the ocean, migration is basically caused by these surface currents. It can be geostrophic, it can be wind-driven currents that happen at the surface, or advection, which happen between the vertical movement between cool and more warmer water, and that create also a wider circulation. So knowing how much carbon is produced by these microbes and how much is being consumed is one of the major challenges today, mostly because the change of this biomass of these microbes is very fast. These microbes can divide, can grow, can reproduce at more than once per day. So if you want to understand the dynamic of this population, you need to observe them at the daily scale. And so the downside of this is that we have very little observation, and most of our knowledge of how these cells grow and how they are consumed are based on laboratory experiments. And laboratory experiments, no matter how fancy you make them, they are never really, they can never reflect the complexity of physical, chemical, and biological interaction that you can see in a natural environment. So 10 years ago, I teamed up with Professor Ambrust to develop instrumentation to be able to measure this population at the relevant temporal scale. So we developed an instrument that we call CFlow. And what CFlow does, it continuously measures phytoplankton. It's single cell measurement. So it does 10,000 cells per second, 24 hours a day, 7 days a week. And so we started 10 years ago. We have now done about 94 cruises, spanning over 200,000 kilometers. That's basically, we went 5 times around the globe, basically. We have 19,000 hours of observation. That's 800 days. And we have characterized about 700 billion Prochlorococcus cells. That's, in terms of data set, it's more than the number of stars in our galaxy. So there are like this, obviously, analytical challenge associated with analyzing these data sets. And so we have created, we have at least done something consistent. Here I'm showing an example, a typical example of the data that are produced by these instruments. On the left side, you see I'm plotting red fluorescence versus light scatter, and every single dot is a cell that was measured by this instrument. Light scatter is a proxy of cell size. The more light scatter, the bigger the cells. And red fluorescence is a proxy of chlorophyll, and that's a pigment that all photosynthetic organisms have. So when we see red fluorescence, we can say, that's a phytoplankton. So, and in this case, you can see like this big green, yellow blob, that's Prochlorococcus. That's how pretty or ugly, depending on Prochlorococcus look under our instruments. But you can see how dominant we see them. We see a lot of them. They are usually 2 orders of magnitude more Prochlorococcus cells than any other phytoplankton in the ocean that we measure usually in this region. We have like some clustering algorithm to be able to separate the different microbial population. And then we have we use some light scattering theory to be able to infer, to estimate cell size, cell diameter from this light scattering measurement. And then with some conversion factor, we can transform cell size to biovolume and biovolume to carbon. So basically, with this data, we can tell how big and how much carbon each of these cells contain. So these are the typical data that we produce. Once we have processed this data, we usually look at hourly average cell abundance of a range of different microbial population. And in this example, I'm showing Prochlorococcus, of course. But we have other population that we can also identify with this technology. So I'm showing here one example, a tiny example of a few days of data sets for Prochlorococcus near the Northeast Pacific. And so we have cell abundance, we have the mean, so carbon quota is basically the amount of carbon contained in each cell. And then we have biomass, carbon biomass, which is basically the product of abundance by the carbon quota. And one thing you will notice, I don't know if you see my mouse, but if you look at the carbon quota, you see this kind of like this oscillation, this diel oscillation. For instance, April 21st, you see the carbon quota increasing, then decreasing, increasing, then decreasing, and then different location, different time, this oscillation dampens or increase at different time. So this is what I was describing to you guys before. Like these cells, they are growing. So you can see their cell size, their biomass increasing, and then they divide. And so you see a decrease in carbon quota. So you can see these repeated cycles of like growth, cell division, growth, and cell division. And so with this diel oscillation, this change in size, we can plug some models to try to infer actually this rate of cell division and this rate of cell growth. So I use a special class of model called a matrix population model. And they are commonly used to estimate demographic rate for measurement of abundance across different life cycle stages. So basically the assumption, this model assumes that the state of a population at t+1, for instance, can be inferred for the state of population at time t and some transition rate that are written as transition metrics, projection metrics. So for phytoplankton, we know what these stages are. The cells can either grow, so they can fix carbon. So it means the size, the size will increase. You will move to the next state, next size classes. You can also divide. So your size class will shrink by half, because cells, when they divide, one cells become 2, so their size shrink by half. And they're also, like any living organisms, they also respire. So they can also lose some carbon due to respiration. And so these models are basically, we are having this size dependency, like for instance, like in the middle, in the top panel, I show you the probability of division given your size. I'm telling you when they are very small, they grow, and at one point when they get bigger and bigger, they are very likely to divide. So these are this type of functional form that we use to infer division rate based on the different cell size. And then the growth is easy to model because we know they're photosynthetic. So the more light, the faster they're going to grow. So that's another one. And we also have carbon loss, and we have some understanding of like how usually carbon loss is a function of carbon fixation rate. So there are some dependency among these stages that we can plug into these models and then infer, for instance, division rate for this population. So here is an example of we apply these models to a big, gigantic experiment, culture experiment, to make sure that the model is able to produce some accurate estimate of growth rate, where we grew Prochlorococcus these cells alone, just with plenty of nutrient and enough light, and we just follow them over 48 hours to look at this change in cell size over time. But we also added to this culture various amount of virus that we know can kill these cells, and other grazers that are also can eat very well on Prochlorococcus. And so there are basically two conclusions I want you to draw. Is like, if you in a control, and when you have a moderate amount of grazers and virus together, the matrix population model can do a decent job estimating growth rate. The truth is what is in red. And so that's what we were measured directly. We know at what rate these cells were dividing. And so that gives us confidence that the model under moderate grazing and virus pressure can do a decent job. But we also know that when there is actually a lot of grazers and a lot of viral lysis, our model breaks down and the accuracy is getting really poor. This is not too surprising, given that we don't have any, when the cells are infected by virus, they are really different. They are not behaving normally. They are kind of like zombies. And so we don't have a zombie parameter in this matrix population model. So that would be an interesting next project to maybe add virus and grazers into this matrix population model to see whether we can still accurately predict. I want to highlight that when we add like this high grazer and high virus, these are really grazing pressure that we rarely see in the environment. So it's really to test kind of the limit of the model. But overall, it gives us confidence that under normal conditions, a matrix population model can be used to estimate growth rate in nature. So that's my third part I want to talk to you about. Using this approach, we have a lot of seaflow data and we have these models, and so we can start looking into how this population, how Prochlorococcus is adapting to different changing conditions. And the two conditions I'm really focused on are temperature and nutrients concentration. The reason I'm looking at these two parameters is because these are the two main parameters that are known to be really affected in the future due to climate change. And so here I'm showing kind of like a canonical view of what we think the future will look like, the future ocean will look like. So we have, of course, like different scenarios of like whether if we keep having this high greenhouse emission, which so far we are on track to be in this high greenhouse emission scenario, unfortunately. If we were keeping this under this doom scenario, we see that climate models predict, on average, the ocean being 3 degrees warmer than what it is today. And then because the temperature of the water is changing, I was mentioning to you like a lot of temperature is also what drives the mixing of the ocean. So the deep water going into the surface, and the deep water are the one that brings a lot of nutrient to the surface. These are these movements that fuel the phytoplankton that live at the surface. So because you have less differential in temperature, you have less mixing, and therefore you will have a less nutrient concentration at the surface. So and the current scenario that we have, if temperature increased by 3 degrees Celsius, we will have a 50% reduction in nutrient concentration. So the question is now, how is Prochlorococcus or these microbes are going to adapt, are responding to this change in temperature and nutrient concentration? And this is kind of a very common way of modeling the growth rates of microbial population as a function of nutrient concentration and temperature. There's basically two terms. One is describing how the growth rate of this microbial population change with nutrient concentration. So we can see like this type of functional forms where at the beginning, a small change in nutrient concentration can really trigger a sharp increase in growth rate and then slowly plateau. The more food you give, then the less they're gonna increase their growth rate. With temperature, there is this based on also some experiment in the lab, few observation in nature. We think that the growth rates of phytoplankton, of microbes, increase exponentially with temperature. So here you can already say like this competitive effect, where if you have a warmer temperature, they're going to grow faster, but if you have less nutrients, they may grow slower. So the question is like how these two competing effects will affect the growth rate of these microbes. And so these are the growth rate that we measure in natural populations. So there's two here type of data set that I'm showing. One is the red dots, which are we're estimated using like a traditional method that we call dilution experiment. Is basically we are it's a method that was developed in the 80s where you basically dilute your sample enough that you don't have any more grazers or virus in the water. So then the phytoplankton grow alone happily, and then you can just monitor the cells over time to see how the rate at which this population divide. These are really painful experiment to do. These are really tedious, difficult experiment to do. So that's why we have so few red dots actually. And usually these numbers are associated with large uncertainties, but unfortunately, we don't have a certainty reported in these data sets. In green are the growth rates estimate from the matrix population model from the seafloor data. So all these greens is basically what we have been collected in the last 10 years. We have where we have seen Prochlorococcus enough, and when we could observe Prochlorococcus for at least 24 hours. We have more data sets, but a lot of the data points were suspicious. So because we have to look at the population over 24 hours. If during these 24 hours, we are entering different water or something happened, then we cannot really trust this measurement. We have to kind of follow the same population over 24 hours. And that's kind of like on a ship moving 24 hours is hundreds of miles distance. So in this long distance, you often may encounter different population or different water mass. So for this reason, even if we have 800 days, we have here 150 days a data point for which we have confidence in our estimate. But it's together with this old data set and this new data set, it gives us kind of a coverage, a decent coverage, at least latitudinal coverage of Prochlorococcus in the surface ocean. And we see kind of like right now a bit of a messy signal where we see basically variability, high variability in division rate as we move towards lower latitude. And then we also see this rate. So 1.5 per day, this is the highest we've seen. 1.5 per day means that cells were dividing twice a day. So they grow and then they divide twice in a row. And so yeah, that's pretty fast considering where they live. They live in this very blue water where there is very few nutrients. So they're able to divide that fast because they are very small and very efficient in uptaking nutrients. So here are the two, if I then look at the relationship between these rates and either nitrate concentration that we measure at the same time that we make these measurements, or versus division rate versus temperature, we don't see a lot of correlation. We don't see a relationship basically between nitrate concentration or any other nutrient concentration that you measure for that matter and division rate. And that's kind of to be expected because as I said, they are so small. These cells are so small that they just need a tiny amount of nutrient to be able to be happy. So even if this concentration of nitrate are really low, for them, for the smallest photosynthetic organisms, this is still plenty. So to be expected, we don't see nutrient doesn't seem to be a big driver in division rate. On the other hand, if I look at temperature versus division rate, we see kind of like for the maximum growth rate, we see an exponential shape, which is kind of like I was discussing, like what we should expect for our microbes. When we, of course, in this, like we cross many different ocean, many different conditions. So we're not talking here about the same type of Prochlorococcus. There is a diversity of different cells there, and some of them grow really well from 15 to 25 degrees. Other will be really doing well between 20 and 30 degrees. So there is a collective of different type of Prochlorococcus that are shown here in this plot. Here, I'm showing the top quantile of this growth rate, the maximum growth rate that we observe for these different temperature. Once at the low temperature, from 15 to 25, we see that our observation matched pretty well what the exponential increase of growth rate with temperature. Let's follow very well what people have been the current assumption that many modelers are using for modeling phytoplankton in the ocean. But above, from 25 degrees and above until 30, we don't see anymore an exponential increase. Instead, we see that growth rates are actually kind of like in a plateau. We should, for instance, at 27 degrees, we should see cells growing at 2 per day, and we have not seen them yet. We have not observed such a high rate. So, but of course, in this ocean that are really warm, this tropical ocean, they also have very low nutrients. So a logical explanation will be, well, you don't see high division rate at high temperature because they are maybe limited by something you have not measured. There may be not enough nutrient for them, and that's why you don't see this high rate. So to investigate this a bit further, I'm adding here on top of the growth rate that we measure in natural population. I'm adding here in blue estimate of division rate measured in isolate. So these are cultures of Prochlorococcus grown under artificial conditions in the lab. So we give them plenty of light, we give them plenty of nutrients, and we measure and we acclimate them at different temperature range, and then and this is what I'm showing here. And so one thing that strike is like even in culture, so it means when they have plenty of nutrients and good light conditions, we also do not see, we were not able to see them dividing at 2 per day at, for instance, at 30 degrees. Instead, we actually see that we don't even succeed to grow them as well as what we see in the field. That has been known for quite some time. These tiny cells, these Prochlorococcus, are actually very sensitive to metals. For them, metals like iron is very toxic. And so in the field, on land, in Seattle, in your lab, it's very hard, when you grow them in culture, it's very hard to prevent this toxicity to happen. And there is also, because they're very tiny, their cells are very tiny, they also need sometimes the help of other bacteria or other microbes to grow well. So in the environment, they have all of they need and sometimes more. And so we were not too surprised to see that actually they are able to divide faster in natural environment than what we do in the lab. But if we assume that we are still doing a decent job in growing them in the lab, not at their max, but still okay, we never succeed to see these very, very high rates of division in culture or in the environment. So this led me to revisit a little bit this assumption of exponential increase of growth rate with temperature. And instead, I've added, I'm arguing that that temperature may be detrimental to Prochlorococcus. Like any species, we have an optimum temperature. We have like a sweet spot where too cold, or sometimes can be too cold or too warm. So you can imagine that in the place in the ocean we have been, or the isolated 360 grow in a lab, we have not yet found a Prochlorococcus that actually like it hot. So there is always this option. But if you really focus on the data we have in hand today, we see no evidence that Prochlorococcus can follow this exponential growth rate with increasing temperature. And so when I apply this new model, where I'm adding for this inhibition, this detrimental effect of temperature and growth rate, we can see that we actually, so this in red is this inhibition, this detrimental effect of temperature, where, for instance, like Z is the temperature at which growth rate deviate from the exponential model. So for instance, in the plot I showed you before would be kind of like around 25 degrees. And W is the width of the terminal niche. So in this case, the width will be between, they can go between 10 and 30. So they have a 20 degree span of a width of a terminal niche. So if I use this new equation, this improved model of growth rate, our accuracy of division rates improve a lot. So in white, these circles is using the model without this detrimental effect. And so you can see between these models here are predicted by, if I know the concentration of nutrients and I know the temperature, therefore I can, and if I know these parameters here, therefore I can estimate growth rate for any given temperature and at any given nutrient concentration. So this is what I'm predicting division rate based on nutrient concentration and temperature. And I compare this estimate with what I observed with the seafloor data, or with this incubation experiment, or with this incubation experiment. So I compare it with the division rate that were estimated in natural population in the ocean. We can see that the new model, the black dots, are more represent better what we see in nature. So so the next question that I have is, okay, so whether I'm assuming an exponential model, I can see the consequence. Why should we care? Okay, why should we care whether it's exponential or whether there is kind of like this detrimental effect at very warm temperature? Well, here I'm using this equation, so this equation here, mu, and I'm adding this equation, these parameters to the equation I showed you earlier. The change of the biomass is driven by growth, so mu, by mortality, these are driven by these grazers, these virus, and by migration, these ocean currents. So I put these equations, these bricks, into this like a numerical model, so a fancy physical model of the ocean, where we have atmospheric pressure, we have currents, wind currents. We have also nutrient concentration. We know how much nutrient are in these different water parcels. And if I add these Prochlorococcus in these models and I let them drift with the currents, and then I can measure how much biomass, I can predict how much biomass I think we will find there. And so here, the difference between the exponential and inflection model, so whether I assume exponential growth rate with temperature or there is a detrimental effect, has actually a surprisingly high effect on what we think are the biomass of Prochlorococcus in our current ocean. By 35%, like with 35% less biomass than previous estimates. only by just changing this functional form, whether it's exponential versus where the temperature can be detrimental. So this has actually a pretty strong effect. So, we were saying before at the beginning of my talk that there is 3 octillion cells, or maybe there's only 2 octillion cells. It's still plenty. There's still a lot of Prochlorococcus. It's still going to be a very important microbes in the ocean. And on the right, I'm showing our projection of the biomass of Prochlorococcus between the difference between what we see now and what we expect to see in the future. And so you can see how in the current model, the exponential model, we were basically assuming that Prochlorococcus will increase in the future. And that was in very good agreement with the study I showed you at the beginning of my talk, when they were predicting, just based on this correlation between temperature and abundance, a 30% increase in Prochlorococcus by 2100. But here, this model with exponential was in very good agreement with this. But now if you add this inflection model, so if you assume that temperature may be detrimental, in this future scenario where water will be warmer, you can actually see that Prochlorococcus is not going to be increased by 30%, but it's actually going to decrease by 30%. So that's a 60% discrepancy with our previous estimate. It's not going to increase. It's the opposite. It's going to decrease by 30%. So that's, of course, that makes me sad for Prochlorococcus. But there will be still going to be growing happily deeper in a deeper, cooler water where there is not this extreme temperature. And they're also going to be able to grow now, expand their range, their latitudinal range to higher latitude. So we can see in red, Prochlorococcus will expand. We're going to maybe see some of them in Puget Sound by 2100, who knows? So there is some, not everything is equal. There are some places where we're going to see more of them, in other places, especially in this very warm water, where we're going to see less of them, unfortunately. But again, who cares? Okay, if you're not a lover of Prochlorococcus, one may say, well, okay, Pro is going to disappear, but something else is going to take over. So that was the second thing I'm looking at here. Here, I'm looking at all the phytoplankton combined. So I have, in a previous slide, I'm just looking at one class of phytoplankton. I'm just looking at Prochlorococcus, and that's it, nothing else. Here, I'm looking at a model that has Prochlorococcus, that has larger, the charismatic phytoplankton that I mentioned to you earlier. There is also grazers, virus. There is a bunch of so much more complex environment. And I'm looking at how much this change in Prochlorococcus model in growth rate will affect the entire ecosystem. So primary production is how much carbon is produced by all of the phytoplankton combined in the ocean. And we see that just as changing this one thing on one species, on Prochlorococcus, just assuming that when it's really, really warm, they're not going to be able to grow fast, this actually has an impact on the global carbon cycle. So we see a 10% less primary. We predict, we think that today we are overestimating primary production, because actually it's 10% less than what our current model are predicting. And that's because even if Pro may not be growing as fast, they may be replaced by cells that are tiny cells that are slightly larger. And so because they're slightly larger, they are not as efficient in uptaking carbon with such a low amount of nutrient. They are not as good as Prochlorococcus at fixing carbon. So it's not like Prochlorococcus die and nothing will take over. No, there are cells that will totally be happy with Prochlorococcus demise, but they're not as good at them. So overall, the primary production will still is lower. And so again here, when I'm then using the same approach and then projecting primary production by 2100, we are actually finding that primary production is not going to increase in the future. before we thought, well, maybe the large cells won't be happy, but Prochlorococcus, the small cells will be very happy. They will be increasing by 30% by 2100. So no worries, there will be phytoplankton. The ocean is still going to be increasing its productivity. Our current prediction says that actually, no, they're not going to increase as much as we thought. 2%, we've probably, meaning there is no increase in primary production by 2100. So it's not a great news. I'm really hoping that I'm wrong, actually. I'm really hoping that there may be some kind of Prochlorococcus, a diversity, a type of Prochlorococcus that I have not yet measured, because we have not measured everywhere in the ocean. So I'm hoping that that's the case. But again, if you really stick to the data that we have in hand, this is not what we can say right now. With only focusing on the data that we have, I think that unfortunately, the ocean is not going to be more productive in the future. So they won't be able to mitigate some of the carbon dioxide that we produce. That's kind of like going along the line again about how we really have to act now, and that the ocean are not going to come to the rescue. Anyway, I'm sorry to have this more like doom scenario. And again, I'm really hoping to be wrong. So to wrap up a bit, if I three things that I would like you to walk away from this talk is the name, Prochlorococcus. Next time you hear this name, I hope you're going to make the connection. It is the most abundant phytoplankton in marine ecosystem. If they produce a lot of oxygen, a lot of biomass, we are here because of them. We are made of some of the carbon that they fix for us. But despite its success in the current ocean, Prochlorococcus is sensitive to changes, and particularly to temperature. So our hope that Prochlorococcus will come to the rescue, will be able to offset some of the change based on the data, the new data that we have now, thanks to the 10 years of research we have done. Doesn't suggest that Prochlorococcus may be the magic bullet, the magic solution to this change, to the global warming. And so that's, right now, that's the conclusion I draw from data that I have, is that ocean warming is unlikely to lead to an increase in Prochlorococcus biomass and primary production. So stay tuned, hope to be wrong. And with this, I'm gonna take some question if there is any.
What a lot of work. Thank you, Francois, for describing your work today. I was thinking a couple of questions, but you answered them during your talk. So I'm like, what can I ask Francois next? And I was thinking maybe you could elaborate a little bit more on you talked about Prochlorococcus finding new regions, like deeper perhaps, or somewhere else, as a function of the species, of course. And that will interact with temperature conditions and so on. But how about the mixing and the nutrient concentrations?
So you mean like how
the combination of the two will affect the different species in the way you envision they might migrate to, perhaps?
Yeah, so I'm looking here in kind of like the really the surface layer of the ocean. All of this work is really focusing on this surface layer, and this is where basically when you have like low nutrients, a lot of light, it's very hot, and you have low nutrients. So it's kind of like a very terrible place to live, actually, if you think about this. So a lot of the phytoplankton actually are usually the sweet spot is a little bit deeper when it's a bit cooler, less light, and they have more nutrient, because nutrient come from the bottom of the ocean. So if you are at the bottom, you're the first one to get use of these nutrients. And so I think there, Prochlorococcus in the future, they will be still very happy. There's nothing alarming there. They will be thriving because they don't need a lot of light, and it will be cooler. That's the main reason they will be cooler, so they're going to be able to grow there. But we were discussing this with Professor Ambrose before. It's also like a lot of if we just focus on some place in the ocean, like the tropics, a lot of islands, for instance, tropical islands, rely on fishing. And there, you know, even if you have current, and maybe if we start to see Prochlorococcus in Puget Sound, awesome, there will be more fish. But in a lot of places, it's going to be really affected. A 10% decrease in biomass for us doesn't mean much, but trust us, for an ecosystem, 10% in lower productivity is huge. So you will see this will affect basically the whole ecosystem, the food chain. There will be less fish.
Yeah, that's right. That was my follow-up question. Like, how is that going to affect the other species?
Yeah.
We do have a question. Given the huge population size, there must be enormous genomic diversity. Lab experiments especially will not sample this, and it is, of course, hard to model. Thoughts on how this might play out?
That's a very good question. So this may hold some some of the things that we think maybe we don't have. So you're totally right. We don't have this in a model, and it's very hard. We are still right now uncovering the diversity of Prochlorococcus, and it's huge. It's really big. There's really a lot of different Prochlorococcus, and all have their own set of skills. Basically, some can grow really well there, but so it's always possible, as I say earlier in my talk, that we have not just sampled a particular Prochlorococcus that may thrive in this very warm environment. So it is possible, but we are starting to look at now at the fact that they are so small. So physically, they are the smallest cells. They are, if you look at they are so packed that you cannot put much stuff inside the cell. So because they are so small, if you divide fast, then you need to store things to be able to get ready for cells to divide. So we think that just the physical size itself, it's kind of, that's why we think that Prochlorococcus may not be able to increase its growth rate at very warm temperature. So we think it's not just diversity. Or if you have different genes, you may be able to grow faster, or you have an enzyme that can actually do well in this condition. We think it's also like just the physical dimension of the cell itself that would prevent these organisms to actually grow fast, to keep increasing its growth with temperature. So, but again, like, evolution and always keeps surprising us. So it's totally possible that there is something in the making that we are not just sampling, and in the decade or so, we're going to start to see Prochlorococcus dividing fast in warmer temperature. But I think it's now the time to say, like, if you just look at what we have now, and if we have to make policy based on, we have to drive policy based on what we know now. And what we know now is not great. I will not, recommend to let's keep going as we do, because at some point evolution is going to come to the rescue and is gonna help us. I hope that's going to come for sure. But again, as scientists, we should really just describe the data that we have in hands. And right now, that's the conclusion I come to with the data that I collected. But again, we have not sampled everywhere, so we're going to keep sampling, go on these cruises. But so, yeah, I hope that answers the question.
Well, thank you, François. I'd like to wrap up with a question for the future. What are your next plans?
Well, I mean, my next one is keep going in this warm region, especially in winter in Seattle. No, like seriously, because I think this warm region in the ocean are the future. They are the future. So we should really focus on going with research vessels in this warm region and look for these rare Prochlorococcus with this set of genes that may be able to make it. I think we should really, with my student, we're gonna focus on this warm region. Next year, we're gonna go back to this around New Zealand, Fiji Islands. It's beautiful, by the way, but especially to really measure this, do we see some sign of stress? We're going to look at the DNA. We're going to look at how these cells are doing to see whether to understand them better. So my next step will be definitely going more into the molecular realm, looking at actually the gene expression and see how actually are they responding to these changes, not just looking at the size and this growth rate. There may be some sign of new population or new diversity that may be able to take over in the future. So that's what I'm going to try to do next time. Yeah.
Well, thank you. Well, we're looking forward to, seeing you back at the data science seminar with better news and signs of adaptation to the warmer temperatures.
Yeah.
Thank you for your participation in our seminar, and
You're welcome.
yeah, bye. We'll see you next time.